Welcome to the Enzymology for Recombinant DNA Technology series. This is episode number 2. As this is the last slide, we will discuss about these type of enzymes. Now, the ligase is finished. Now we comes to the nuclease. So, nuclease are broadly defined as the enzyme that degrades polynucleotides. They comprise a large family of enzymes that shows diverse reactions and substrate specificity, such as DNA specific known as DNAs and RNA specific RNAs, nucleotide sequence specific or non-specific, either way, double strand specific or single strand specific, isonucleolytic or you can say endolytic, or generating 3' phosphate or 5' phosphate, many other things. Many polymerases, including reverse transcriptase, also exhibit some sort of intrinsic nuclease activity with widely varying reactions and substrate specificity, which makes the rational division of nuclease even more complex. Historically, nucleases have been divided into DNAs and RNAs, but many nucleases degrade both DNA and RNA, blurring the division based on sugar specificity. For almost ubiquitously, existence of large variety of nucleases suggests that functional roles of nuclease could be diverse, yet none of these suspected in vivo functions have been clearly identified. Nevertheless, our knowledge of the structural and functional characteristics of individual nucleases has dramatically increased. The accumulated knowledge has led to wide and important applications of nuclease in the analysis of nucleic acid structure and nucleic acid protein interaction. Nucleases are now an important tool in nucleotide sequencing and various other recombinant DNA techniques. So we will discuss about these 5 types of nuclease: DNA nuclease, DNA RNA, RNA endonuclease, RNA nuclease, single stranded specific nuclease, and DNA exonuclease. So first we discuss about DNA endonuclease. DNA endonuclease is also known as DNase1 and first isolated from the bovine pancreas. It is an endonuclease that cleaves both single-strand DNA and double-strand DNA to produce primarily 5' phosphate dinucleotides and 5' phosphate oligonucleotides. DNase1 degrades double-strand DNA in a sequence non-specific manner. This enzyme requires divalent cation such as calcium ion, magnesium ion, and magnesium as a cofactor for full double-strand cutting activity. This enzyme having molecular weight of 30.5 kd and made up of 4 glycoprotein components, DNA is A, B, C, and D. The substrate specificity for this enzyme, before we going to the substrate specificity, the international definition, your definition is written here. Now we comes to the substrate specificity. Substrate. The first we comes to the substrate preference. So all those single strand DNA are substrate and double strand DNA are 100 to 500 times better substrate for this enzyme. Single strand DNA of 40 nucleotide in length are cleaved faster in central region beyond to the 10 terminal nucleotides. This distinction disappears as substrate length increases. Now we comes to the product profile. DNase1 digestion gives primarily dinucleotides up to the 60% and trinucleotides up to the 25% and other nucleotides as a final product. The products have 5' phosphate and 3' hydroxyl terminal double strand cleaves of DNA in the presence of magnesium ion results in staggered and with 2 to 4 nucleotides in the 3' direction. Now it comes to the substrate size. The smallest substrate for DNase1 is the tetranucleotide carrying 5' carrying 3' phosphate group. Now we comes to the nicking activity in the presence of magnesium ion. Under certain suboptimal reaction condition, DNase-1 produces nicks rather than a scission of both strands. Here I want to tell you that if we want to make nicks on the DNA, then we have used to this magnesium ion. Otherwise, other cations, calcium and magnesium ion, we can use for DNA chopping. Now it comes to the nicking activity in presence of the ETBR. In presence of high concentration of ETBR up to the 20 to 80 microgram per ml and with the 2 millimolar magnesium chloride, DNase-1 introduce a single nick to the plasmid cccDNA. Now we comes to the application of this endonuclease. This is very important thing. As non-specific nuclease, DNase-1 is extensively used in molecular biology and recombinant DNA technology. The following some examples are here. I'm giving you for using various strategy for. Number one is the non-specific degradation of the DNA. DNA-1 is commonly employed as a non-specific nuclease to eliminate contaminating cellular DNA in protein, RNA or protein precipitation. DNA-1 is also used to remove the template DNA after in vitro RNA transcription using sp6 or T7 RNA polymerase. Now we come to the DNA labeling by nick translation. As I told you earlier that magnesium ion we have to use for nick production. So DNase-1 introduced nicks into the double-strand DNA under certain suboptimal reaction conditions, such as using magnesium ions. The nicks serve as a priming site for E. coli DNA polymerase 1, which by virtue of its 5' to 3' exonuclease activity removes nucleotide from 5' site of the nick and the same time synthesize a new strand from the 3' side of the nick in a process called nick translation. In the presence of one or more radioactive DNTPs, labeled DNA suitable for hybridization can be generated by the nick translation. Now we comes to the random insertion of mutagenesis. This is very important thing. DNase-1 introduces a single nick into the supercoiled plasmid in the presence of high concentration of ETBR. Now we comes to generation of DNA fragments for deoxy sequencing. DNase-1 digestion in presence of manganese ion produces double-strand break in the DNA. The average size of the products from partial digestion is inversely proportional to the amount of enzyme used, and the distribution, such as lambda DNA, sponge, is smooth and unimodal. Therefore, DNA-1 can be used to generate random DNA fragments for both shotgun cloning and, most importantly, non-random cloning. Now, another application for DNA is the footprinting. DNase-1 footprinting is one of the powerful techniques used to study the insertion of the ligands with DNA in solution. Here, the ligands is used for the proteins and drugs. When there is at least 10-fold sequence specificity, the technique provides direct and immediately information about the location of protein or drug binding site in the DNA sequence. The extent to which a footprint represents protein binding varies with the protein DNA complex studies because the rates and thus pattern of the DNA cleaves are affected by the both the width of the minor groups and the flexibility of the DNA. Now we comes to the DNA RNA endonuclease. This endonuclease first isolated from the bacteria Staphylococcus nuclease is also known as micrococcal nuclease, and it is calcium dependent phosphodiesterase which cleaves both DNA and RNA to yield 3' phosphomononucleotides and 3 phospholigonucleotide end products. In short form, it is written as SNase, and it is a monomeric enzyme with both endonucleolytic and exonucleolytic activities. Now we comes to the international unit definition that is written here. Now we directly comes to the substrate specificity. So first is the polynucleotide. The denatured DNA means the single strand DNA is preferred substrate over the duplex DNA. Denatured DNA is digested by SNase sevenfold more effectively than RNA, and no inhibitory products are accumulated during the reaction. Now we comes to the cleavage preference. With denatured DNA, the order of cleavage appears to be nearly random. In native double-stranded DNA, however, some bonds are preferentially cleaved. The cleavage preference at a 5' side of the A or T is estimated to be 30 times greater than that of the G or C. Although the AT-rich regions are cleaved preferentially, the transit single strandedness of the such regions is not only the recognition feature. The most rapid cleavage site tends to occur in stretch of the more than the three categories A and or T residue. Nevertheless, long stretches of DNA containing both A and T have been observed not to be cleaved by the enzyme. Now we comes to the another specificity of this enzyme, dinucleotides. Dinucleotides means only two nucleotides are the substrate with this distinct order of preference. I mean, first is T, then A, then C, then G for the basis of the beta position, but the little base specificity in the alpha position. Now we comes to the mononucleotide. It's the synthetic substrate with the basic structure of the nucleotides are generally cleaved by the SNase with the release of RP or nucleotides. The size and the nature of the R group are relatively unimportant as long as the diester bond is present. Now we move to the application part. So its applications are: number one is the preparation of 3 phosphomononucleotides. The relative non-specific activities of SNase can be used to generate 3' phosphate mononucleotides from a wide variety of DNA and RNA substrates. The 5' hydroxyl of the 3' phosphate mononucleotide can be efficiently phosphorylated by the T4, P, and K. And next is the removal of non-specific DNA and RNA. This is a very important thing. SNase is frequently used to prepare eukaryotic cell-free extract from the rabbit, reticulocyte, lysate, wheat, gram, and many other things, which support the in vitro translation of a wide variety of viral prokaryotic and eukaryotic mRNA into the protein. Brief digestion of the endogenous mRNA by the SNase reduces the background of the in vitro translation to minimum, leaving the rRNA and tRNA largely intact. Now we use as a sequence-specific hybrid nuclease. By fusing the relatively non-specific SNase with specific oligonucleotide, the SNase is transformed to hybrid nuclease which can sequence-specific cleavage of single-strand DNA and RNA. Now the next is the probe for the protein binding or distortant region of the DNA. SNase is a small protein. Yeah, it's very small. It's about 15 to 17 kD compared with the DNase one. DNA is, as I told you earlier, is more than 30 kD kilodalton. which is mostly widely used in the DNA footprinting. SNase recognized as a single strand for DNA double helix, whereas DNase one interacts with both strands in the minor groups. As is as an essentially single strand specific endonuclease, SNase relies on the breathing motion of the duplex DNA to make cleavage. Furthermore, the cleavage at G and C residue can be subsequently slower than the A and T residues. Now we move to the now it comes to the ribozymes, the RNA endonuclease. These constitute a superfamily of nucleases that catalyzes the hydrolysis of phosphodiester linkage in RNA. For the majority of RNAs, the catalysis involves the formation of nucleosides, 2' 3' cyclic phosphate intermediates, and generation of products with 3' phosphate group. These enzymes are present in all organisms, including bacteria, yeast, plant, animal, and in almost all tissue and body fluids of the mammals. Some RNAs are secreted, that means extracellular, and probably suggesting their major roles in the digestion. Other RNAs, so-called cytoplasmic or intracellular RNAs, are usually found in latent form, complex with protein inhibitor, and yet they share some structural characteristics typically of secreted RNAs. Within a single cell, a number of functionally distinct RNAs can be found. There are hun- there are uh, hundreds of RNAs isolated from enzymes, but we have to read here only three. RNase A, which is isolated. This is also known as pancreatic ribonuclease because of this firstly isolated from the pancreas. That next is the ribonuclease H, which is also hybridase because it degrades RNA DNA hybrid. And then next is the ribonuclease T1, which is also similar to the RNase A. So first we discuss about RNase A. So RNase A is this, as I told you earlier, this is also known as bovine pancreatic ribonuclease. Is the most thoroughly characterized of all the RNAs. It is the first enzyme and only the second protein, actually first is the insulin, whose amino acid sequence has been determined. The proteolytic cleavage of RNAs by substitution results in the two fragments, S peptide and S protein. When the two fragments are held together by non-covalently interaction, full RNase A-like activity is restored. This enzyme has other species of this enzymes are RNase B, C, D, which are isolated from the pancreatic secretions. And the substrates suitable for this enzymes are the polynucleotide substrate, natural or synthetic single-strand RNA is preferred substrate for this one. And dinucleotide, in dinucleotide substrates, the second base can be replaced by a wide variety of bases such as deoxynucleotide, modified ribonucleotide, and nucleotides with attached alkyl groups. Mononucleotide and derivatives are also its preferred substrate. Pyrimidine, like pyrimidine 2' 3' cyclic mononucleotides are it's a substrate also. Uridine 3' phosphate aryl esters are also its substrate. And this enzyme activity in alcohol is very high concentration of alcohol. This enzyme is also active in aqueous methanol is hydrolyzed by 3' CMP and methyl esters. The products are alcoholysis. The alcoholysis is the reversal of the cyclic phosphate forming steps in the most effective with the glycerol followed by the ethylene glycol, menthol and methanol and ethanol. Now we comes to the ribonuclease H. As I told you earlier that this this enzyme is also this enzyme is also sorry for I have to uh, tell you about the applications of RNase A. So first is the RNA sequencing. So RNA sequencing, it's like a DNA sequencing. This RNA sequencing is one of the technique where RNase A cleaves RNA by pyrimidine bases C and U. Therefore, partial digestion of RNA samples results in fragments terminating with C or U. The fragments are then separated by phage polyacrylamide gel electrodes to form ladder. When the pattern of ladder is exposed to those of the base specific RNA such as RNase T, G specific, and the RNA sequence can be read from the sequence ladder. Next is the preparation of DNA material. RNase is frequently used to degrade RNAs in plasmid and other DNA preparations. Following digestion, RNase A is removed by the phenol chloroform extraction and the DNA is purified by the ethanol precipitation. See the fission from and removal of unhybridized region of the DNA, RNA. Sorry. The fine substrate specificity of RNase A can be utilized to cleave the single-stranded regions of RNA in DNA, RNA or the RNA hybrids. Next is the biochemical applications. So, in addition to all the above applications in recombinant DNA technology, RNA has various biochemical and genetic tools, which I discuss here, as a helix destabilizer. Because of its ability to bind both DNA and RNA at the superphosphate backbone, RNA A also functions as a melting of helix destabilizing protein. And it's also used for the model for protein structural studies. RNA is very small, structurally and functionally well characterized enzyme. Therefore, it is an ideal model for the study of not only the folding pathway but also the relationship between the protein structure and function by means of various molecular biological techniques, including the site-specific mutagenesis. Gene expression and protein digestion is also RNase is also excellent model for studies in gene expression, protein stability, and this degradation. Now we move to the RNase H. Now it comes to the RNase H, which is also termed as a hybridase because it degrades RNA strands of DNA RNA hydroduplex. Initially discovered in extract of calf thymus as a contaminating enzyme during the purification of RNA polymerase, RNA H has since been found almost everywhere in lower and higher eukaryotes as well as in the prokaryotes. The international unit definition is written here. Now it directly comes to the substrate specificity. The typical substrate for this enzyme is that specifically degrades the RNA moiety in the DNA RNA hybrids. That means it always degrades the RNA from the hybrids. The product profile, this enzyme digestion are 5' phosphate oligonucleotides of 2 to 9 nucleotides in length. It chop up the RNA into the 2 to 9 nucleotides. Now it comes to the direct cleavage of RNA DNA junctions. This RNA previously in vitro cleaved the 3' to 5' phosphodiester bonds that links RNA and DNA. On modern substrate, DNA extended tRNA forming a duplex with the DNA strand. However, E. coli enzyme was shown to the cleave phosphodiester bond, not the DNA RNA junction, but on nucleotide away from the it. So now Now we come to the application part. The first is the synthesis of cDNA. The This enzyme plays an important role in cDNA cloning strategies. Following the first and cDNA synthesis using a reverse transcriptase, a number of strategies are available for the synthesis of the second cDNA synthesis. Now we come to the removal of acyclic tail. The poly A tail of eukaryotic mRNA can be removed from the RNA by the RNase digestion following the hybridization of the mRNA with the oligo DT. Oligo DTs, yes. Next is the quantitation of the poly A tail in mRNA. Poly A tail mRNA can be selectively quantitated from the total mRNA of the mixtures with the rRNA. The method relies on the two enzymatic steps. Number one, polymerase catalyzed labeling of the 3' termini of all RNA species in the presence of its and long phase analog chain terminator, and this reaction added one or most a few nucleotides. And next is the RNase digestion of the total sample in the presence of oligo DT, releasing radio-labeled poly A tails exclusively from the RNA molecules. Next is the specific fragmentation of the RNA. The observation that RNA annealed to the short synthetic deoxynucleotide can be site-specific cleavage by the RNase that serve as the basis of the diverse amplification of RNase H in RNA mapping or sequencing. Next is the hybrid arrested translation by the antisense oligonucleotide. The small antisense oligonucleotide directed inhibition of gene expression or translation has emerged as a powerful technique for studying the control of the gene expression in vitro as well as in vivo. The oligonucleotide directed cleavage of the destabilizing of the target mRNAs by RNase H has been found to be, at least in part, mechanism of the antisense oligonucleotide directed inhibition of the gene expression. The last is the detection of RNA containing the double-strand DNA. This is very important. The specificity of RNA can be used as to detect the presence of RNA in DNA structures. A covalently closed circular DNA that contains RNA can be converted to the release form, which after denaturation produces a single circle and a single stranded molecules. Now we comes to the last part of a ribozyme, the T1. T1 actually is a very specific nuclease, guanine specific endonuclease, which is isolated from Aspergillus oryzae. It catalyzes the hydrolysis of phosphodiester bond on single strand DNA, producing 3' phosphate mononucleotides and oligonucleotides that end with the G, means guanine. Unit definition is written here. We come to the substrate specificity, base and sugar specificity. RNase T1 cleaves the single strand RNA especially at the G bases. However, the base specificity is not absolute. After a long incubation period at high concentration of enzyme, cleavage at other bases occurs a measurable extent. Now it comes to cyclic mononucleotides. RNase T catalyzes hydrolysis of cyclic nucleotides. Dinucleotides are also the substrate, and tri and oligonucleotides, although dinucleotides are the substrate for RNase T, one catalytic efficiency is optimal with oligomers 3 or more nucleotide long. Trans-esterification reactions with trimeric substrates are significantly different from the reactions with the dimeric substrate. A dramatic attenuation of the pH-dependent, so no discrimination for nucleotide leaving groups and all that. So what are the, now we comes to the next thing, this application of RNase T1. So very short applications, very RNA sequencing. It's actually for the radio-labeled. I mean, when the three radio-labeled phosphorus RNA is partially digested with RNase T1 and separated by the phase, the latter yield information of the ordering of the fragments terminated with the G residues only G residues. It's a site-directed cleavage of the RNA. The cleavage of single-strand RNAs by T1 can be directed to a specific site by the following steps. These Now it comes to the RNase T1 fingerprinting. It's like fingerprinting. It's a fingerprinting. Complete digestion of RNAs with RNase T1 yields oligonucleotides ending with the guanine nucleotide. It in high salt concentration, which tend to promote the double helical structure. RNase T1 selectively digests single-strand RNA while leaving the double-strand RNA intact. Now we comes to the site single-strand specific endonuclease. These in nucleus hydrolyze both DNA and RNA in a highly single-strand specific manner. These nucleases degrades single-strand polynucleotides as well as the single-strand regions in double-strand polynucleotides. Depending on the species of origin of the enzyme, nuclease can be more or less specific suited for the double-strand DNA and RNA. Single-strand RNA, just like nuclease S1 and being nuclease. So first we discuss about the nuclease S1, which is also known as S1 nuclease. It is an endonuclease highly specific for single-strand polynucleotides. This is actually a glycoprotein of about 35 kilodalton and is isolated from the Aspergillus oryzin. This enzyme cleaves DNA and RNA both. So the unit definition is written here. Now we come directly to the substrate specificity and product profile. Nuclease S1 hydrolyzes both single-strand DNA and single-strand RNA. It is 2 or 3-fold more active on single-strand DNA than on single-strand RNA. The products of digestion are 5' mononucleotides and a small amount of 5' di and 5' oligonucleotides. According to the cleavage preference, this enzyme shows little or no sequence specificity as long as single-strand polynucleotide exists in a state of a random coil. And single-strand specificity, nuclease S1 can recognize and cleave single-strand regions of duplex DNA or RNA, non-hydrogen bonding regions. Non-hydrogen regions are the anticodon groups in tRNA. Partially denatured or locally altered regions containing nicks, gaps, and mismatch are cleaved but with a varying efficiency. The last is the circular DNA. S1 Nuclease S1 can introduce nicks into the circular superhelical double-strand DNA, which is later converted to the linear molecule. When the content of superhelical unit gets lower than the 30 to 40% native DNA, a circular DNA molecule undergoes a transition to a negative supercoil, producing a localized unwinding of the helical base pair and rendering them to hypersensitive to the nuclease S1. Now we move to the gene nuclease. This, also known as nuclease MB, is a single-strand specific endonuclease similar to the S1. Nuclease MB is also a glycoprotein with a similar molecular size, almost, and its definition is here. And now we directly come to the substrate and product. While nuclease MB catalyzes the hydrolysis of single-strand nucleotide, the digestion products are 5' mono and 5' oligonucleotides. Single-stranded RNA are hydrolyzed at approximately equal rates as a denatured DNA. Cleavage preference, nuclease MB exists in cleavage preference For 80 rich region, the lambda double-strand DNA is also cleaved most rapidly in centrally located 80 rich region. Cleavage as mismatching sites like nuclease S1. This nuclease cleaves the region of single and double mismatches very inefficiently, whereas three or more unpaired nucleotides are cleaved efficiently at high enzyme concentration. Next is the single strand specificity. This nuclease act on single strand DNA versus double stranded DNA varies widely depending on the types of DNA and the reaction condition. Just as for 37 degrees centigrade under optimal condition, 30,000 fold for T4 DNA and 65-fold for the CRAP 80 rich region, less than 2-fold for synthetic 80 rich region, and no detectable hydrolysis with G or C homopolymers. Endonuclease associated with the 3' phosphate. This enzyme causes an intrinsic 3' monophosphate activity which dephosphorylates A' mononucleotides. Now we come to the application of both of these endonuclease. The first is the cleavage of single strand terminal. Duplex DNA with either 3' or 5' protruding ends can be made blunt end by the treatment with nuclease S1 or nuclease ND. The reaction is usually carried out or below 25 degrees centigrade. Depending on the reaction condition, fully blunt end duplex molecules may coexist with equal or greater amount of imperfect duplexes with +1 or +2 nucleotides. And this another application, important application, is opening of the hairpin tubes. During the synthesis of double-strand DNA complementary to RNA, the second strand of DNA can be synthesized using E. coli DNA polymerase by self-priming of the first DNA strand from the 3' terminal hairpin structure. Nuclease S1 has been instrumental in cleaving the hairpin loops to generate double-strand DNA suitable for subsequent ligation into the clonic vector. This method results in a low yield of cDNA and has the drawback of sometimes losing the critical information at the 5' end of region of the starting the RNA. Next is the RNA secondary structure analysis. This used for the structural fingerprints, as I told you earlier. Assay of the DNA RNA hybrid and RNA DNA mapping also. Now we move to the DNA exonuclease. DNA exonuclease means the enzyme that cleave the DNA from the terminal ends. This enzyme is a monomeric multifunctional enzyme. Very short enzyme is about 35 kilodalton and catalyzes the hydrolysis of at least 4 different types of phosphodiester bonds in double-strand DNA. The main enzymatic activity of this exo3 is the 3' to 5' exonuclease activity that carries out the successive reads of 5' mononucleotide from the 3' ends of double-strand DNA. The unit definition, international unit activity is written here. And now we directly come to the substrate specificity, double-strand specificity. EXO3 is a double-strand DNA specific 3' 5' exonuclease with sequentially released 5' mononucleotide as products. Double-strand specificity. Now we come to the base picture. Exon 3 hydrolyzes DNA with varying structures that at similar rates, no difference actually. Extent of digestion, partial digestion of the double-strand DNA produces a duplex with a protruding 5' case. As the digestion proceeds, the length of the duplex region becomes progressively shorter. The remaining base paired region eventually becomes too short to sustain a bihelical structure and separates into the single strand that are resistant for the further digestion. Now the processivity and synchrony. Exotherm digests double-strand DNA in either processive or a non-processive manner depending on the temperature and divalent ions. And, now we come to the altered or modified substrate structures. Exotherm can neither hydrolyze alpha thiophosphate containing phosphodiester bond nor bypass them. Now we come to the application part of this enzyme. So, sequencing, DNA sequencing, this enzyme is used for this. So this enzyme is used for by by two methods, by this digest template and generating deleting mutation. This next is the oligonucleotide direct mutagenesis. This is also used in mutagenesis, site-directed one. Next is the polymerase repair synthesis. Limited this enzyme double-strand DNA leaves a 3' hydroxyl terminal that can be used as a starting site for the prime DNA synthesis by DNA polymerase. Such partially digest DNA molecules are excellent strategy for assaying DNA polymerase activity. The partial digest DNA molecules are also useful for the localizing mutagenesis, DNA labeling, this type of things. And this enzyme is also used for the analytical methods such as detection of AP sites, AP endonuclease activities, and this mapping of mutation sites in the DNA. So that's it. Thank you for listening.